Hello, my name is Andrew Reich, and I am in Biology 510 at Grand Canyon University. Thank you for watching my presentation of connecting cellular concepts to physiology with a focus on vision. About this presentation, there'll be parts of it where I want it to be interactive. So if I'm going too fast or you want to rehear something, since this isn't live, feel free to pause and rewatch that segment. I broke this presentation into three main parts. The first part is the objectives, and then the importance of this presentation. That's followed by the science content focus, and we're going to be talking about G protein coupled receptors, more specifically their role in phototransduction and the production of vision in the human eye. And this content is geared towards an undergraduate biology major who has already taken an introductory biology course and is now in a molecular biology class. And finally, we will finish up with a discussion of a systems approach or the pedagogy involved in this presentation. All right, so two objectives. These first two objectives are for biology students. The first objective is that students should be able to discuss the general anatomy and physiology of the human eye. And second, students should be able to describe the role G-coupled protein receptors play in cell communication and apply their knowledge to a novel situation. Two more objectives, these ones focused on future teachers of science. First, learners should understand that the importance of how teachers instruct abstract scientific processes. And two, learners should be able to integrate the larger picture into their teaching of abstract scientific processes. So, why am I making this presentation? Well, first off, we have to acknowledge that scientific concepts can be abstract, especially when you're looking at molecular biology. We can't see these with the naked eye. We can't even see them with a light microscope. We have to use an electron microscope, which in order to do that, you have to have a dead organism, which means you can't see the live functioning of it happening. You can only kind of see still shots. In addition, the electron microscope doesn't give us a clear picture of what everything looks like, because sometimes we'll have parts that are inside other parts. So what we have to do is we have to use a process called X-ray diffraction, and all those techniques are not something that most undergraduate students are familiar with. So this type of concept is something that can become difficult for them, so we have to acknowledge that. Second, the traditional educational models encourage compartmentalized learning. So you learn about photosynthesis, you learn about cellular respiration, but a lot of times they never connect those two together and see how that relates to the whole ecology in general. So molecular biology, you talk about different macromolecules or metabolites, but you never put multiple of those together to create a function of the organism. Right? And then finally, and less important, that the funding for science education has decreased over the last several decades. We are in a current upswing in it, but nowhere near what was or percentage-wise was being put towards science education in the late 50s, early 60s, when the Russians launched the Sputnik satellite and the space race began. Right, so we will start this presentation off with a general review of the basic eye anatomy. All right, so I break the eye into kind of two main parts. I break it into an anterior portion and a posterior portion. We can see that the posterior portion is significantly larger than the anterior portion, and we can even see like this big, thick white layer that goes all the way around the posterior portion. This white area is called the sclera, and it's fibrous connective tissue, and it's what gives the eye its shape and its strength. So the eye is a hollow organ about 2.5 centimeters diameter, and it's filled with a very viscous solution. And so it doesn't have any internal support structure, so we have to rely on the outside structure, and that would be the sclera. Right. Then the yellow part here, this is the retina, and the retina is what we're going to be we'll be focusing on in a little bit, so we'll come back to that. In between the retina and the sclera is the choroid, and that has a lot of vascular tissue that supplies both the sclera and the retina with nutrients and helps remove wastes. All right, let's move over to the anterior portion of the eye. We have this clear layer on the outside called the cornea. The cornea is made up of a lot of neurons, but these neurons lack myelination. So the signal gets transported a little slower than other neurons, but we're able to see through them. If you had all the myelination, that'd be putting a lot of fat. You'd get a yellow glaze at whatever you're looking at. In addition to lacking myelination, the cornea also lacks vascular tissue. So we call it avascular, meaning that there's no blood vessels in there. This is a really cool feature because of all the body parts that you can donate. The cornea, the recipient doesn't have to worry about rejection of the cornea because there is no blood vessels and no antigen-antibody interactions. All right, separating the anterior portion with the posterior portion, we have the iris. The iris is a smooth muscle that controls the size of the pupil. The pupil actually isn't a structure, it's just an opening. And it appears black when you're looking on from the anterior side because on the inside of the posterior portion of the eye, there's no light being generated, so it looks like a void. It's black. Okay. As the smooth muscle contracts, the pupil will get larger, and then as the smooth muscle relaxes, the pupil will get smaller. And then finally we have the lens, and then the lens is controlled by the ciliary bodies that can help focus an image back on the retina. So as I said earlier, the retina is responsible for actually turning light energy into chemical energy. Alright, so what is the function of the eye? Most people, including grade school students, when you ask them what the function of the eye is, they'll probably tell you to see. So the function of the eye is vision. And in humans, vision is actually the dominant sense that we use. We can remember things that we see better than we can remember things that we hear, feel, or touch. And then if you add combinations of those, then you can just enhance the memory of that signal. All right. But in order to see, we have to have some sort of sensory receptor. We know that there's receptors that identify heat, touch, and stretch. So there also has to be a receptor used in vision. And that receptor that we use in vision is called the photoreceptor. Right? And is what the photoreceptor does is it will absorb light energy and convert that light energy into chemical energy, which then eventually gets transported throughout the cell or from one cell to another or even down one cell as an electrical impulse. Alright, so what type of light energy do photoreceptors detect? They only detect visible light. Visible light is in the middle range of the electromagnetic spectrum, and it's organized by wavelength. You've probably seen Roy G. Biv, and that stands for red, orange, yellow, green, blue, indigo, and violet. Red has the longest wavelengths, or the distance between two crests, or two troughs, and then violet has the shortest wavelength. Okay. So the shorter wavelengths have more energy, and you can also increase the amplitude, and we'll talk about that here in a little bit. Alright, so photoreceptors they come in two flavors, or two main flavors. We have rods, which is right here on the left hand, the blue on the left, and then we have cones, which is the pink one on the right hand side. Rods and cones, they're both photoreceptors, but they also have different functions. Rods, they can only detect black and white vision, while a cone, it can detect color vision. In fact, cones can be further differentiated into red, green, and blue cones. All right, back to rods. Rods, they're really good at identifying different intensities of light. So if we go back to looking at the visible light, the intensity would be the amplitude or the measurement of how high the wave is. So this would be a stronger intensity than this one. Right. Cones, on the other hand, they don't really do intensity well, but they're very good at definition and clarity. So if you want a nice, sharp, crisp image, that would be the responsibility of the cone. Surprisingly, rods are a lot more numerous than cones. There's a little more than twice as many rods than cones in your eyes. And of all the receptors that we have, photoreceptors are the most common receptors that we have in our body. All right. One more thing I'd like to mention with rods and cones. Rods are usually found on the peripheral edges of the eye, while cones are mostly located in this little area called the macula, and to be even more specific, it's a part called fovea. Right, so cones are really focused right there, which is just a good place for the lens to focus a lot of light, and then the rods are located in higher concentrations on the peripheral edges of the retina. Alright, so speaking of the retina, we're going to look at a closer image of it. And so right here is just a larger or close-up version of the retina. Here we can see the retina is broken up into 4 parts. We have epithelial cells, then we have our rods and our cones, then we have a layer of bipolar cells, and finally we have our ganglion cells. So those are the 4 layers that we have. Notice again, there are a lot more rods than cones. Another thing to take notice of is that the light actually goes through the ganglia, the bipolar cells, and most of the rods before it actually gets to this little part here at the tip of the rod or the cone where the phototransduction actually occurs. Alright, so again, here is a rod. Right? And if you're going to look at the outer segment, we see if we pull back the plasma membrane, it's kind of packed with these thin pancake organelles, and we call these discs or rod discs. Right? And these rod discs are just like every other phospholipid bilayer. So if you want to go back and review what a phospholipid bilayer is, you can look at the top right image over here. Right? So we have a layer, we have 2 layers of phospholipids with the hydrophobic tails pointing in and the hydrophilic heads on the outside. But what I want you to draw your attention to are these blue things that are also both in and on the one end of the phospholipid bilayer. The proteins that go all the way across, those are called transmembrane or integral proteins, and a lot of those are ion channels or involved in some sort of receptor-mediated communication, and we're going to be looking at one of those in a little bit. We also have a peripheral protein that has a complex that is on the inside or the hydrophobic area of a phospholipid, and also on the outside. And then finally, we just have a surface protein. So we have integral, integral, peripheral, and surface proteins. All right, so going back over to this image where we're looking at the outer segment of the rod, we can see that these phospholipid bilayers in here, they have lots of these orange little dots. So these orange little dots, they go all the way across. So that's going to be a transmembrane or an integral protein. So we're going to learn a little bit more about those in the next slide. All right, so here's a close-up of those orange dots. Now we're showing them as purple, but that is an integral protein called rhodopsin. We can see that the rhodopsin, if you look closely, there's 2 parts. there's the amino acids that makes up the easy thing to see. But if you look closely on the inside, you see a ball and stick model of a molecule, and that molecule is retinol. Retinol uses vitamin A to get synthesized, so we can't make retinol without vitamin A. That's why you always hear eat your carrots to get your vitamin A for your vision. More specifically, retinol, it kind of comes in two stereoisomers. You can have it as in the cis retinol, or you can have it in the trans retinol. And that's going to be important when we start doing our signal transduction. Right? So just by looking at this computer-generated image, we can see in the back that this would be our light source, and obviously rhodopsin has something to do with that light. Alright, so now we're going to be looking to integrate some of our new knowledge or learn something new and apply that to what we've already learned in past biology classes. So if you could just look at that image on the left-hand side of your screen, we have the rod cell, and then we have a very close-up of the rod cell. When we're looking here, we can see that there are two phospholipid bilayers. This one where my pointer's at, this is the cell membrane. And then you have another phospholipid bilayer. This is going to be the rod disc membrane. All right. On top of the cell membrane, we have our side, or the outside of the cell, or the extracellular space. In between your plasma membrane and your rod disc membrane, we have the cytoplasm, and then down here we have the inside of our disc. And we're not going to really be doing too much discussion about this space. We're going to mostly be talking about what's going on inside the cytoplasm. All right. If you look back at the plasma membrane, we have a sodium ion channel. More specifically, this is a chemical gated sodium ion channel. So it requires a chemical to be bound to it in order for it to be open. So here we can see that the sodium ion channel actually has the chemicals bonded to it right now. So in the resting state, the sodium ion channel is always open, which means sodium ions can have a net influx into the cell due to the difference in concentration gradient outside and inside. And then last thing to remember, that these are sodium ions. So sodium ions do carry a positive charge. Alright, so step one. So first thing we have to do is we have to absorb a photon of energy. We have to absorb the energy. So that is done on the rhodopsin, or inside the rhodopsin molecule, more specifically by that cis retinol molecule. So if you remember the cis retinol, that's what's inside the opsin. So this molecule inside here, it will absorb a photon, and when it absorbs the photon, it actually will change from a cis retinol into a trans retinol. And that's just kind of, it's going from a bent formation into a straight linear formation. When the retinol is in the trans isomer, then the retinol, it will dissociate with the opsin protein, and it'll actually leave the complex. When that happens, the opsin protein itself goes under a conformational change, so the opsin molecule will then change its shape. Right. So that was step one. All right, step two, we have to activate the G protein complex. So once the opsin molecule undergoes that conformational change, it will bind to and interact with this G protein complex. All right, and inside the rods, the G protein that we're going to be specifically talking about is transducin. So there are many different types of G proteins, and there's a different G protein in between rods, and then even there's different G proteins in each one of the three colored cones. But in the rods, the G protein is called transducin. All right, so one thing I want you to notice with this G protein here is that there are actually three parts to it. And we call these subunits the alpha, the beta, and the gamma. So three different parts. Another thing to notice that the G protein, it is a peripheral protein. So it is on the outside of a phospholipid bilayer. So this G protein will always kind of be attached to the outside of this phospholipid bilayer that makes up the rod's disc. All right. So we can activate the G protein complex by changing a GDP into GTP. So when you have these three subunits here, one of the subunits will be holding on to GDP, and that'll be the inactive state of the G protein. But what we're looking for is we're looking for that GTDP to get changed to GTP, and that is done once opsin binds to the G protein. Right. Okay. So once the G protein has exchanged its GDP for a GTP, the alpha subunit, it'll actually start to move away. And so it'll actually separate from the beta and the gamma subunits, and it will move over to this third protein. And this third protein, we call this phosphodiesterase, or PDE for short. Sorry. All right. So, what is phosphodiesterase? Now we have to go take a look. All right. So phosphodiesterase, we know it's an enzyme because it ends in ASE, as all enzymes do. But specifically, phosphodiesterase, it breaks down cyclic GMP or cGMP into GMP. Remember that sodium ion channel that was in the plasma membrane we talked about earlier? The ligand that it requires is cyclic GMP. If you remove cyclic GMP, then the sodium ion channel will close. So what phosphodiesterase does is it turns this cyclic GMP into just GMP, and so then the sodium ion channel will close. When the sodium ion channel closes, the sodium ions cannot diffuse into the cytoplasm anymore, which will then affect the membrane potential of the plasma membrane, because we're no longer bringing in these positive discharges anymore. So let's look at what happens in the membrane potential. Alright, so at the resting state of a rod, the membrane potential is around negative 40 millivolts. When phosphodiesterase turns cyclic GMP into just GMP, the sodium ions stop coming in. So without those positive charges coming in, the voltage will drop, or the potential will drop. So the membrane potential will drop from negative 40 millivolts to negative 65 millivolts, and that happens in a relatively short span of time. Right. Remember, this is only happening when a photon is being absorbed by a cis retinol, and then it'll be a quick thing, and then it goes back. So it will slowly go back over time. The way that you can extend this out would be to keep exposing the cell to light. So if you keep the light source on, it will stay exposed to it like so. Alright, so that change in membrane potential, it will actually get passed down the cell by adjacent areas. So although the rod is not myelinated, it can slowly pass that hyperpolarization down. Let me go right back to this. When we're going more negative, we call that hyperpolarization because we are getting more negative. Right. So the hyperpolarization impulse, that will travel down the rod, or remember, going into the interior of the eye, and it will pass through the cell body, and finally will reach the synaptic ends, or the synaptic region. When that hyperpolarization wave reaches the synaptic ends, there are there's a bunch of voltage-gated calcium ion channels located in these synaptic endings, and they respond to this change of voltage by closing. And so when the voltage-gated calcium ion channels close, synaptic vesicles cannot exocytosis or go through exocytosis and release neurotransmitters. So therefore, when the voltage-gated calcium ions close, then fewer neurotransmitters are released from the synaptic endings. So therefore, that membrane potential will close calcium ion channels. And why is it important that we're closing these calcium ion channels? Well, those calcium ion channels allow the neurotransmitters to be sent from the photoreceptor layer into a bipolar neuron, which then sends a signal to a ganglion cell. So we essentially are slowing down the transfer of information, or even essentially stopping some of the transfer of information from the rods and the cones to the bipolar inner neurons. When that happens, fewer action potentials, AP, action potentials in the ganglion cells is the result. All right. One thing to know, that in the absence of light, rods and cones are always on. So when there's no light, rods and cones are always on. Okay. When light comes in, it will temporarily turn off the corresponding photoreceptor. So for our rod, when light came in, it turned off. And so these fewer impulses get sent to the bipolar cells, to the ganglion cells, and eventually get sent to the optic nerve and to the brain. And the brain reads this absence of information and builds a picture of that in our head. Alright, so that was kind of a lot of information going from a photon of light being absorbed by a cis retinal all the way to an action potential being sent to our brain. So let's just do a little bit of review. So look at this image on the left hand side of your screen, and this is inside the rod, but what portion of the rod are we looking at? You can pause the video if you have to think and go back through. If you said this was showing the rod disc membrane, you'd be correct. Right? We can see we have our rhodopsin molecule with the retinol on the inside. We see our G protein. In this case, since we're in the rods, we're looking at transducin. Right. We see that the G protein, the alpha subunit of the G protein, can dissociate after GTP or GTP replaces GDP, and then we see the third enzyme, phosphodiesterase, that was responsible for hydrolyzing cyclic GMP down to GMP. All right. So looking at this image, where is the cytoplasm located? Again, you can pause it if you need time to think. All right, if you went back to the slides in the beginning, you'll notice that the G protein, it is a peripheral protein, and so being a peripheral protein, it's always on one side of the membrane, and it's on the side of the membrane that faces the plasma membrane. So if the plasma membrane is somewhere over here, then we know that this area here is the cytoplasm, and then we know this is inside the disc. All right. So why can the proteins in this image move freely around the membrane? So if our G protein is coupled to this receptor here, and we can even see this alpha subunit is free to move around this membrane, how can that be? If you said that it's due to the nature of the phospholipid bilayer, you'd be correct. The tails of the phospholipids allow the membrane to be fluid, specifically allowing molecules on the ends or part of it to move from one spot to the other inside the membrane. Alright, so 3 more review questions of the process we talked about. First, I'm going to start with the second question right here. what molecule is responsible for absorbing a photon of light? If you can look inside there, that's what we were looking for. What is the name of that guy? If you said cis-retinol, you'd be correct. So the cis-retinol is inside of the rhodopsin complex. All right, so a little harder. Can you list 5 proteins and the role that they played in this process? Again, pause to think and answer. All right, so the first protein that we talked about was rhodopsin. More specifically, it was the opsin protein of it. All right. Once the retinol absorbed the photon of light, it made the opsin molecule change shape, which activated our G protein. Our G protein in this example was transducin. Remember, there's many different types of G proteins. We just looked at transducin, but they all kind of function the same way. All right, the alpha subunit of this G protein, it turned on phosphodiesterase. Phosphodiesterase hydrolyzed cyclic GMP into GMP, which then caused the sodium ion channels to close. So that was our fourth one. Remember, these ones were ligand gated based on the cyclic GMP. Oopsies, that was spelled ligand gated. Right. And so we need one more. Remember, after the sodium ions stopped moving into the cell, we caused a hyperpolarization wave that traveled down the rod, eventually made its way to the synaptic ending of the rod, which closed voltage-gated calcium ion channels. Okay, so those are the 5 proteins we discussed. All right, and when we were talking about going from the closure of the sodium ion channels to the closure of the calcium ion channels, we talked about the hyperpolarization wave, and so that would be the answer to the third question. Right. Okay, so that was the content, our first part, or the second part. So now we're going to go into the third part of this presentation and look at the pedagogy style that we used. Right, so we use a systems approach, and when we were examining the systems, or how the different parts of the whole interact, we use a constructivist philosophy. There are 2 main constructivist philosophers or psychologists, Piaget and Novak. Piaget believed in assimilation and accommodation, and so assimilation means to Piaget that when you are learning, you're kind of constructing a tree in your mind, and then when you learn a new piece of information that just extends a branch, that's assimilating. You're growing your tree of knowledge. It just fits nice into your base of knowledge. On the other hand, accommodation to Piaget would be if you had your tree already set up and you got a new piece of information, but it didn't fit anywhere in your tree, so then you would have to rearrange your whole tree in order to fit that one piece of information into it. And so that would be accommodating. As teachers, when we present material and the students are forced to accommodate it into their mindset, this is a longer process and it takes more work. Assimilation, on the other hand, is an easier, quicker process. So our goal is to try to assimilate knowledge into the pre-existing concepts that students already have, unless their concepts are riddled with large misconceptions. Then the misconceptions will have to go through accommodations. But our goal is to assimilate new knowledge instead of rebuilding new knowledge every time we teach it. All right, so that was Piaget's theory. And then Novak's theory is very similar. He uses different words. Instead of saying assimilation, he talks about meaningful learning. And instead of accommodation, he says rote learning. So what is the difference between meaningful learning and rote learning? Meaningful learning is going to be the image on the left, and rote learning will be the image on the right. So when a student learns something, he keeps it in his head and it's compartmentalized. But then when they learn new facts, they can link things together. And as they create these linkages, it reinforces the concept of the old concept and the new concept, therefore creating a longer lasting memory. Rote learning, on the other hand, if the student has his prior knowledge here, you're going to be adding new knowledge bases, but you're not making many connections. And because you're not making these connections, these individual pieces of information are easily lost. So therefore the goal of the teachers is to make meaningful learning and assimilate it, instead of making students change their mindset or not connect any of the information. So how did we do that in presenting the content? Well, we took the prior knowledge that the student learned in prior classes of the structure of the eye, the structure and function of a plasma membrane, and then they know about seeing and vision, and we showed them a chemical pathway that connected all those together. So again, we're looking at a systems approach, or how we can connect different related terms. All right. So in this one, we looked at a cell communication pathway to the physiology of a cell. So we started at the molecular level, where we had a protein, rhodopsin, that absorbed a photon of light, and then it followed a chemical pathway that eventually led to an organelle becoming dipolar or hyperpolarized. So when the cell membrane became hyperpolarized, we were changing the organelle, and now we are looking on a larger scale. So we're no longer looking at the individual molecules, we're looking at larger portions. Eventually, we got to the cell part, when we had the rod communicating with the bipolar cell, and the bipolar cell communicating with the ganglion. We connected different cells. Eventually, that led to different tissues. So your rod is part of the peripheral nervous system, and then your brain will be the central nervous system. So when you get that ganglion going into the brain, you're going from one tissue type to another tissue type. And the organ that we examined was obviously the eye. We looked at, you had to have an opening and a means to control how much light came in. That was done by the iris, a smooth muscle. We had a lens that focused the light. We saw that the retina is where our photoreceptors were located at, but the distribution of photoreceptors changed based on the location of where you're at in the retina. And then finally, we came out with a function. In this case, we came out with sight. So we started out on the molecular level and ended on the functional level. All right, so the focus was placed on connecting different topics that the student has learned about in the past. So therefore, we were introducing the G protein coupled receptors, but we're reinforcing concepts that were previously learned to the student. So that's systems, bringing everything together. Hopefully that creates more meaningful learning and help the student assimilate knowledge instead of reconstructing or accommodating that knowledge. All right, thank you for listening to my presentation. There are 10 references on the next 3 slides if you would want to pause the video and look at any of them. Some of those references are focused on the science content and learning how G proteins or the eye function was determined. Several of the other ones were based on educational pedagogies and why scientists should do, or why educators should teach in a certain way. Again, my name is Andrew Reich, and again, thank you for listening to my presentation.